Symptoms may include fever, fatigue, dry cough, and some people may experience aches and pains, nasal congestion, runny nose, sore throat, and diarrhea. It is said that the viral respiratory infection is spread by direct contact, such as touching an infected person or the surfaces that that person has either touched or on which large virus containing droplets expired by the person have landed. The virus can stay on surface for several days. Quarantine is a period of isolation after being exposed to an infectious disease. Quarantine can last up to 14 days immediately following the day after exposure. It also depends on how severe the exposure is and how sick that person may be. Megan and Kat's family is in a 14-day quarantine because one of Megan's coworkers were tested positive for the coronavirus, so they have been exposed. I do think that Megan and Kat are at risk of catching the Rona because Megan's coworker was tested positive for the virus and Megan shares an office with her. Kat also stated that she has been in that office, even if it was only for an hour, she was still exposed. DNA is a double-stranded micromolecule that is composed of nucleotide. RNA is a single-stranded and short polynucleotide. One human infected virus that is DNA based is herpes. Glycoproteins are proteins found floating around in the membrane of cells. One of their functions is to act in cell to cell recognition and binding molecules. The coronavirus have glycoproteins in their membranes because membranes represent both an obstacle and aid to replication. Virus encoded structural and non-structural proteins that have membrane binding properties. These proteins are used by the virus to help with infection and replication in host cells. The cell membrane consists of two layers of phospholipids, which also forms a phospholipid bilayer. The phospholipid bilayer consists of two sheets of phospholipids arranged tail to tail. The hydrophobic tails associate with one another, forming the inferior of the membrane. Hand soap is made up of molecules called surfactants that have a hydrophilic end and a lipophilic end. Surfactants like lipids in the lipid bilayer of enveloped viruses like SARS-CoV-2. These surfactants can wedge their way into the lipid bilayer syphilic end first, causing the virus to break apart. First, you have attachment, where the plague attaches to the surfaces of the host. Next, there's penetration, where the viral DNA enters the host cell. Then you have biosynthesis, where fake DNA replicates and proteins are made. After you have maturation, when the plague particles are assembled. Last, the cell release new phages. The steps of SARS-CoV-2 infection are different from lytic infection because SARS-CoV-2 is an RNA-based virus. So instead of attaching to the surface of the host, SARS attaches to the host cytoplasm. An RNA-based genome results in increased infection rate because the RNA polymerase makes complete copies of the genomic RNA to be packaged into new variants and transcribes specific sections to the mRNA as templates for translation from the original infecting RNA. The central dogma of biology describes a two-step process, transcription and translation, by which the information in genes flows through DNA, RNA, and protein. The central dogma describes the process of transferring genetic information inside a cell. DNA passes the information to messenger RNA, which then moves to the ribosome, where it's used as instructions to build a protein. But the coronavirus does not use DNA. The process starts with RNA passing the information to mRNA. Antibiotics are medicines that help stop infections caused by bacteria. They do this by killing the bacteria or by keeping them from copying themselves or reproducing. Coronavirus is caused by a virus. Antibiotics only treat bacterial infections, not viral infections. Antiviral drugs are prescription drugs that fight against viruses in your body. Antiviral treatment works best when it is started immediately after illness. Antiviral do not destroy the host because it inhibits replication. The antiviral drugs that Megan identified would not work for SARS-CoV-2 because none of them target RNA viruses, which is one of the main concerns for SARS-CoV-2. One antiviral drug I would suggest would be Vivilavir, an antiviral drug made in China. It attacks the RNA viruses by inhibiting RNA-dependent RNA polymerase. Coronaviruses replicate their RNA genomes using enzymes called RNA-dependent RNA polymerase, which are prone to errors. Scientists say that COVID-19 is mutating slowly, reducing the chance of it changing to become more deadly. If we get an antiviral drug that possibly targets the RNA, it could destroy the virus.